Everyone, today's lecture is about the chemical transformations of food. So during this activity we are going to talk about the chemical transformation taking place during the or during digestion. So here, as you see here, the protein is digested into amino acids, and these carbohydrates are digested into monosaccharides. So before starting, you have to know what is the meaning of chemical transformations. Chemical transformations means a change in the identity of the substance. So the chemical transformation means a change in the identity. So here the protein is transformed chemically into amino acids. So this is a chemical transformation. Why this is a chemical transformation? Because we have a change in the identity. The protein is transformed into a new substance called amino acids. Always the chemical transformation taking place during digestion is to make the food simpler. To make the food simple, and in this case, uh, this is the interest of the digestion. So here the carbohydrate is made up of high number of rings of sugars, and here it's made up of two rings of sugars, and it becomes monosaccharide, more simple, and in this case it is a chemical transformation. So when a protein, for example, is digested into amino acid, yes, in this case we can say that it is a chemical transformation. But if the protein in in large size, it is in a cube, for example, if we cut it into small pieces of proteins as you see here now, so the protein stays what? Proteins. So in this case here, it's not a chemical digestion, but here it is a chemical transformation. So the chemical transformation means a change in the identity into a new substance. A chemical transformation of food takes place during digestion through a process called hydrolysis. So hydrolysis means make the food simpler, cutting the food into simpler ones. So we need water. Always the hydrolysis means uh, breaking down the substances in the presence of water. Hydrolysis. Okay, so hydrolysis means digestion in the presence of water. By using water and digestive enzymes, we will talk later about the enzymes, to break down the complex molecules of food into simpler ones. During chemical transformation, the identity of the food is changed. Okay, so you can see here the protein is digested into amino acids. So this is a chemical transformation. Polysaccharide and disaccharide becomes monosaccharide. The fats are digested into fatty acid and glycerol. So these are chemical transformation, and these are the importance of them is to make the food simpler. نحن قلنا we said إنه we need enzymes to make chemical transformation, and also we need water. So here you can see the enzyme. What is an enzyme? The enzyme is a chemical substance that speeds up a chemical transformation to make the food more simple here يعني, during digestion. So digestive enzymes are chemical substances that speeds up a chemical reaction and to make the food simpler. So here you can see I have a mistake here. So if we want to talk about the role of enzymes, we said the enzymes, they speed a chemical reaction. What do we mean by speed up a chemical reaction? So For example, the protein, we want to digest the protein into amino acids. This chemical transformation cannot be occurred without a digestive enzyme, without enzymes. يعني ممكن تصير بقلب جسمنا بس بعد تقريبا ما بعرف قديش بدها وقت، بعد سنة سنتين 3 سنين ما بعرف. فإذا هيدا الانزيم اوكي بسرع الرياكشن into 10 maximum 10 exponent 14. So the enzymes speed up a chemical reaction، بسرع chemical reaction تقريبا into 10 exponent 14. يعني في some chemical reactions cannot occur without enzymes. فهول الانزيمز هن اللي بيعملوا الاشياء، من دونهم we cannot make a chemical digestion. Here in this diagram here you can see the enzyme and the substrate. The substrate comes into the enzyme بفوت لجوا and it's transformed into products. So the substrate is digested into products. هون مثلا فينا نقول، وهون فيكم تشوفوا كيف الانزيم عم يعمل molecular simplification. وأكيد هيدي transformation from the substrate into products ممكن تصير بس تاخد كتير وقت، وممكن أبدا ما تصير. فهو الانزيمز هن chemical substances that speed up a chemical reaction. هلا عنا we have here a problem in documents A and B on page 18. The problem here, what is the origin of the sweet taste obtained after chewing a piece of bread for a certain time? We have a problem, we have a question, and we want to solve it. So the problem here, what is the origin of the sweet taste obtained after chewing a piece of bread for 15 minutes أو for a certain time. يعني عنا شخص عم بحط بتمه خبز وتركها for 15 minutes، بعد 15 minutes لقى في عنده slightly sweet taste، طعمة حلوة. اوكي فهون بدنا نعرف what is the origin of the sweet taste، منين اجت هالطعمة الحلوة. To solve this problem an experiment is performed in this activity. هلا ممكن شو تكون الفرضية؟ ممكن sweet taste تيجي from saliva، انه عمل chemical transformation of cooked starch. ممكن يقول حدا تاني انه مش من السلايفا، الستارش alone عمل. يعني بدنا نعرف مين عمل chemical transformation، هل هو الكوكت ستارش لوحده عمل transformation، او هل هو السلايفا عمل chemical transformation of cooked starch انتو مالتوز. منين اجت الطعم الحلوة؟ اول شي البريد، what does the bread contain? The bread contains what? Starch or cooked starch. The starch doesn't taste sweet ابدا، الستارش ما طعمته حلو يعني، ف مستحيل تكون منه. The sweet taste comes from maltose which is a reducing sugar. So we want to study if there is a chemical transformation of starch into maltose in our mouth. So we want to check if the saliva is responsible for this chemical transformation or not. بدي شوف هل السلايفا بيقدر يحول الستارش into maltose وهيدا عنده slightly sweet taste. Let's see. Because of that we performed this experiment by putting cooked starch in both tubes. So tube A and tube B have cooked starch. Tube A also contains only a small amount, only tube A contains a small amount of fresh saliva. So the variable factor here is the saliva. Saliva, it's present in A and absent in B because we want to study about the role of saliva and the effect of saliva on cooked starch. بدي شوف هل السلايفا بيقدر يحول cooked starch into maltose or not. Question 1, we want to draw a table showing the experimental conditions of the performed experiment. What are the conditions? Cooked starch, small amount of fresh saliva, temperature 37 degrees Celsius, and the time which is written below the document here. So you have to check it on your book and to read the duration. Here in this table you can see the result of it. Cooked starch is present in both tubes. A small amount of fresh saliva is present in A and absent in B. The temperature is the same and the duration is the same all the time. So here the variable factor is the saliva because we want to see if the saliva can digest the cooked starch into maltose or it can't. And when we want to draw a table or construct a table, you have to write the title and the key. So don't forget to write the title and the key. Identify the variable factor of this experiment. The variable factor of this experiment is the fresh saliva. It's present in A and absent in B. After that, we have another document which shows the result of iodine test of samples taken of each tube at time 0 and after 15 minutes. At time 0, we perform iodine test which gives positive here and positive here. Why positive? Because we obtain dark blue color. What does this color indicate? The dark blue color indicates the presence of starch. So the starch is present in both tubes A and B. Why? Because we have already added starch. فإذا at time 0 we take some samples. بالوقت صفر أخدنا some samples from this tube and from this tube. شلنا من هون شوي ومن هون شوي. ما نحنا we have already added cooked starch. يعني إجباري طالما في عندي starch إجباري يطلع عندي dark blue color indicating the presence of starch. And after 15 minutes هيدا done. After 15 minutes بعد 15 دقيقة من الاكسبيرمنت اللي حطيناهم هون لقينا انه عملنا iodine test and we observed that here we obtain brown orange and here we obtain the same result dark blue color. يعني the starch has disappeared in tube A. يعني هيدا كان في starch بطل في starch كان في starch and it stays present. Why? Because here we have saliva. But here we have no saliva. So the dark blue color becomes brown orange color, which indicates the disappearance of the starch under the action of saliva. بينما هون without saliva there is no chemical transformation. ما بيتحول chemical transformation والستارش هيبقى starch. Okay. So under the action of saliva, what can we conclude here from this experiment of iodine test? نستنتج إنه the starch has already أو the starch is digested under the action of saliva or saliva can digest the cooked starch. بس لشو ما بعرف لشو تحول. إذا بدي أعرف I want to work with Fehling test. Fehling test is a test to check the presence of reducing sugar. At time 0, Fehling test gives blue color. At time 0, blue color. ما في أبدا reducing sugars. يعني ما أنا حطيت starch ما حطيت reducing sugar. فكيف حتطلع؟ يعني أنا إذا بتطلع هون at time 0 شو حطيت؟ حطيت cooked starch in tubes A and B. ما حطيت reducing sugars. فعشان هيك حتطلع هون minus هون minus. ما في reducing sugars. بعد 15 دقيقة تركتهم هول مع بعضهم أخدت samples منهم يعني لقيت إنه هون عطى break red precipitate بال A وهون عطاني blue color. Why? Because here we have saliva and saliva digests cooked starch into reducing sugar. وعشان هيك هون ظهر عند reducing sugar وهون ما ظهر عند reducing sugar لأنه أصلا ما تحول cooked starch. يعني هون as function of time إذا بدي أرسم لتيوب A هيدا الرسم لتيوب A cooked starch حتنزل كميته وحيطلع محلها reducing sugar. فإذا reducing sugar حيعلى والستارش حينزل because the saliva can digest starch into reducing sugar، بينما بالبي الستارش حيبقى 100% والريديوسينج شوجر حيبقى 0% وما حنشوف الكوانتيتي له. فإذا شو بستنتج هون؟ إنه saliva is responsible for the chemical transformation of cooked starch into reducing sugar. فإذا starch is transformed chemically into reducing sugar having a slightly sweet taste. فإذا لما تحول من starch into reducing sugar صار عندي في reducing sugar، هيدا اللي هو عنده slightly sweet taste. عشان هيك if we put a piece of bread in our mouth for a certain time حنحس ب slightly sweet taste. ليش؟ لأنه saliva in our mouth makes the chemical transformation of cooked starch into maltose having a slightly sweet taste. Okay. Justify if the problem is solved by using document C. أكيد it's solved. Iodine test shows that starch disappeared after 15 minutes in tube A with saliva, while the starch stays present in tube B without saliva. While Fehling test shows that reducing sugar is produced or obtained only in tube A with saliva after 15 minutes, thus the problem is solved and the sweet taste comes from the chemical transformation of starch into reducing sugars by the action of saliva. Here حنشوف كيف السلايفا هيعمل chemical transformation of starch. This is a starch molecule which is made up of high number of rings of sugars. هيتحولوا into maltose اللي هو reducing sugar which is a disaccharide made up of 2 rings of sugars. هيكون عنده slightly sweet taste. Simplification of starch molecules into sugars. Saliva allows the gradual breaking down of the complex molecule of starch into simpler molecules. This transformation is called hydrolysis. So this is a hydrolysis or molecular simplification. أكيد we need water during hydrolysis, so you can write here on the arrow plus water. It's caused by the presence of saliva of a chemical substance, the amylase, which is a digestive enzyme. The resulting molecules formed are reducing sugars giving the sweet taste. فهيري إذا كان لي question name the enzyme present in saliva. The enzyme present in saliva is called salivary amylase. So the salivary amylase need water to make the molecular simplification of or hydrolysis of starch into maltose. Thank you.